culture some of our phytoplanktons here at Culture by Science. So, first things first, we work primarily with 1 gallon jugs. This allows for small batch culturing, prevents contamination. We're able to fine tune exactly when we would like to harvest certain phytoplanktons, especially such as the one I'm showcasing today, Rhodomonus, a species that actually prefers cooler temperatures, and so it is very, very finicky in terms of when it's ready to harvest. We have our sterilized water here. Sterilized DI water is what we use, so RODI. And what I will be doing is, as you can see, the 2 culture jugs have evaporated some water during the culturing process. So I'm going to top them off back to where they should be, which is roughly about right here. As you can see, they're still bubbling, so we're able to mix that DI very quickly through the rest of the culture, so we're all at roughly the same salinity. Now what we're going to do is we're just simply going to remove the lid. As you can see, we have airline running into a rigid airline that allows us to suspend all the way to the bottom of the bottle. This prevents settling. It's very, very, very important. Now I'm going to grab my bottle. I'm going to measure out 800 milliliters, which is going to be our starting solution for our next culture. So now that we have 800 milliliters into our gallon jug, here in my hand I have a sample of our F2 Gillard's fertilizer. So this is what we use to feed the starter culture back to full density. So you can find this product on our website, both in 50 ml tubes such as this one, or in the larger 500 ml bottles, 1 ml for every 2 liters of culture volume. So this is usually the method that I hold to ensure that my instruments here are always as clean as possible. I am trying not to touch any surfaces, even though this F2 is actually only used for Rhodomonus. But regardless, we're trying to keep as tidy as possible. You see the syringe is kept in another plastic container. The bottle is sealed shut. The 2 are put back together. Now all we really have to do is top it back up with fresh salt water. This salt water is just like the DI, sterilized. How do we sterilize? Very simply, these bottles are autoclavable, able to tolerate very high pressure and temperatures, but all we're really doing to sterilize our cultures is via boiling water. Coffee makers. So that tumbler that you're seeing there is, what we do is we add deionized water to it. We allow it to percolate for about 45 minutes until the water is boiling hot. Then we measure out the desired salt in grams for each bottle, and then we fill that with the DI water and seal it shut. That creates as sterile as possible the salinity. Bottle is back up and ready. Here it is, percolating. You can see this is a couple of days old now versus the ones that we're splitting. It's certainly lighter. Every single day we will be seeing it double and double and double in density until it's ready to harvest. In this environment here, we harvest every second or third day, but at home in a lower temperature, you probably can expect to harvest, well, with something like Rhodomonus every 4th, 5th day.